HNDT Agriculture second year students. Again, you are welcome to AG 2106 Diseases of Crops and Their Management subject. Under that, you have to learn diseases of crucifers. Under the diseases of crucifers, you have to learn about infectious diseases and non-infectious diseases. Under the non-infectious diseases, you need to learn about clubroot, damping of black rot and cabbage yellows. Then you have to learn about clubroot disease of cabbage. Then causal organism is Plasmodiphora brassicae, and host range is cabbage, radish, broccoli and cauliflower. You can see here cabbage, radish, broccoli and cauliflower. Then we'll go to the symptoms of clubroot disease. There are above ground symptoms and underground symptoms. As above ground symptoms, we can see the chlorosis or yellowing, wilting during hot days and exhibit the stunted growth. Actually, developing plants, we can't see any symptoms, but the plant get older only we can see the symptoms. Then if the disease is severe, it is very difficult to see the heads. That means failure to form heads. Then underground symptoms, if we uproot the plants, we can see the swelling of the lateral roots. Then that is in club shape or spindle shape roots we can see there. Those are called clubs. That's why it is called clubroot disease. Then they are actually galls. The Plasmodiphora brassicae pathogen secrete the growth hormones to produce the more number of cells in the roots. Then what will happen? Then these excess cells, not for the plant purpose but for the pathogen purpose. They use these plant cells as pathogen food. Then therefore we can see the club shape, a large number of cell growth there. Then but ultimately what happened? It is very difficult to transport the water. Therefore we can see the above ground symptoms like yellowing, wilting, stunting like that. Then we can't expect the proper growth of plants. Then ultimately we can see the malformed root growth and eventually roots will rot and ultimately total plant will die. Here you can see the above ground symptoms, mainly yellowing of plants in different stage. You can see here, you can see wilting also. In this picture also you can see the yellowing of matured plants. You can see well. Here you can see the wilting of plants. Here you can see the wilting of plants. Those are the above ground symptoms. Here you can see another above ground symptom, stunting of plants. This is the healthy plant, this is the stunted plant. Then these are the healthy plant, these are the stunted plants. Here you can see the underground symptoms, swelling or malformation on the main lateral roots. You can see club shape or spindle shape, enlargement of the lateral roots you can see here. Then large number of cell growth happen on the lateral roots. These plants are somewhat mature, but it is very difficult to form the heads here. It's very difficult to form the heads here. It is another symptom of clubroot disease. Actually, this is transmitted by contaminated transplants, animals, surface water, runoff, contaminated equipment and irrigation water. Then the pathogen can survive in the field or in the soil for many years as resting spores until finding the susceptible host. Then actually clubroot disease favor the warmer conditions and soil temperature between 20 to 24 degrees Celsius is better for them, and also they favor the moist conditions for the spreading the disease, and also pH around 5.7, that means slightly acidic level is preferred for the disease. In the spring, resting spores in the soil germinate and produce zoospores. Then zoospores with the moisture, with the drainage conditions, it will go to the healthy plantation. Then these zoospores can swim through the moist soil and enter the host plants through wounds or through root hairs. Then we will go to the disease cycle of clubroot disease. Then we will start from here. Then these are the roots of infected cabbage plant. You can see here club roots here. Then inside these club roots, there may have large number of Plasmodium pathogen. Then after disintegrating the club roots, what will happen is resting spores may come out. Then with the favorable condition, resting spores may germinate and produce the zoospores. Then zoospores can enter into the plants through wounds or through the root hairs. Then root infection can happen. Then there are high cell growth here. Then Plasmodium may congregate inside of these cells and invade into other tissues, and other cells also spread. And sporulation may happen inside of the plants, and inside of the plants, after rupturing this the particular sporulated place, then zoospores emittent, zoospores formed in sporangia, still they are in the host. Then these zoospores are released through the pores. Then this zygote may again infect the healthy tissues. Then and also Plasmodium invade the other cells of the roots also. Then we can see multinucleate Plasmodium here. Then here happen hypertrophy and hyperplasia. Then hypertrophy means they can increase the size of the cell, and hyperplasia mean they can increase the number of cells. Then both hypertrophy and hyperplasia happen, and due to that continuous cell growth and the number of cells also will be increased. Therefore we can see the club roots, enlarged cells in the lateral roots we can see. Then fungus like pathogen filled cells of cabbage roots. Then again they will release the resting spores into the outside. Like that, this disease cycle complete. Actually this pathogen is not a fungus, that is called slim mold, but at last it is come under the protist, not come under the eukaryotic, and it come under the protist. Here also you can see the disease cycle of clubroot disease very simply, and this is the infected crucifer. Then rupture in the infected crucifer. Then what will happen? Resting spores coming out. These are the resting spores in soil. Then these resting spores can produce the biflagellate zoospores. Then zoospores can enter into the plant roots and through the root hairs or wound, and can enter into the plants. Then Thereafter, the secondary infection can happen. Then Plasmodium invades the cells, and they are multinucleates, and they causes the ball formation. And again, we can see the infected crucifer here. Like that simple disease cycle you can understand by seeing this. We'll go to the management practices of clubroot disease. Actually, good sanitation practice is very important because in the disease cycle characteristic I told you, they may have pathogen in the soil and crop debris and all. Therefore, it is very important to maintain the sanitary practices. The use of disease-free planting materials, and also we need to provide the well-drained condition because with the moisture conditions, the spores can enter into the plants and do the infections. Then treat nursery beds and field with sulfur dust as a soil drench. Then it is very important to control the disease. Then keep in the soil at a slightly basic level because you all know at the slightly acidic level favor the disease, and therefore we need to produce the somewhat neutral range or basic range. Therefore we need to add agricultural lime, and we need to apply 2 to 4 tons per hectare, and we can raise the soil pH. Then totally we can control the disease with that. Then as well as the integration of crop rotation, and dip the seedlings in mancozeb solution before transplanting as seed treatment. And also avoid continuous cultivation of crucifers crops because the pathogen may be in the soil. The resting spores of the pathogen may be in the soil for many years. Therefore, it is very difficult to control this disease if we grow the same crop in the same field. Therefore, avoid continuous cultivation of crucifers crops. Then it is very important to use of resistant varieties. Therefore, no problem with the clubroot disease. Then again, we are not teaching you the damping of crucifers because the disease is same as in brinjals. Now you have already learned that. Next, we'll move on to the black rot disease of crucifers. Then actually this is one of the most destructive disease of cabbage and other crucifers. The causal organism is Xanthomonas campestris. Actually this is bacteria disease. We'll see the symptoms of black rot disease of crucifers. Then this disease can happen at any stage of the growth. Then actually there are characteristic symptoms. Then we can see the yellowing at the leaf margin, which expands into the characteristic V-shaped lesions. I can show you the symptoms later. Then actually this is the characteristic symptoms. Yellowing of leaf margins with the V-shaped lesion. Then bleaching of veins also can be happen along the leaf margins, and it will extend towards petiole and the main stem. And the bacterial infection becomes systemic. How it becomes systemic? The bacterium can enter into the veins of the plant and spread into the cabbage head also. Then thereafter we can see the yellowing, wilting and drying of the leaves. And ultimately we can see the defoliation. Then if we get the typical cross-section of an infected stem or petiole, it will show the black ring due to invasion of water conducting vessels. Then if we cut the head also, if we get the cross-section of head also, we can see this cross-section of infected stem, the black ring due to invasion of the water conducting vessels. Then here you can see V-shaped yellow portions. Nicely you can see here V-shaped yellow portions here. See here, V-shaped, V-shaped. Then actually the bacteria enter into the plants through the hydatodes. Here you can see hydatodes. Then through the hydatodes or wounds, bacteria can enter into the plants. The hydatodes are there at the end of the margins of the leaves. That's why we can see the bacterial infection symptoms at the leaf margins. Then after getting the cross section, you can see the water conducting tissues blocked with the bacteria. Therefore, we can see internal vein blackening. That is the black rot symptoms. You can see very nicely here. We'll go to disease cycle, characteristic of black rot disease of Crucifers. Then pathogen thrives in warm, wet weather. Then it will spread from plant to plant by splashing water, wind-blown water droplets, and by workers or by animals moving from infected fields to healthy fields. This bacteria can spread rapidly during transplant production in greenhouses or in seed beds and could be spreading long before any symptoms are observed. Then at very beginning it is very difficult to observe the symptoms with very young cultivation, but later on we can see the symptoms at the same time. The bacterium can infest seed also, then infect young seedlings as they emerge. The pathogen can also survive in cruciferous weeds such as yellow rocket, shepherd purse and wild mustard, as well as in crop debris in the field. We will go to disease cycle of black rot disease of crucifers. Then we will start from here. These are the infected seeds. Then infected seeds themselves produce the disease. Then it will convert into the infected seedlings. Then infected seedlings, we can see the infection in the leaves and in the vascular tissues of the leaves. Then therefore we can see the V-shaped lesions on the leaves. Then thereafter we can see the systemic infection of plants because bacteria progress into the vascular tissues. Then thereafter, then again the seeds can infect and again come to the infected seeds, or the pathogens survive in the plant debris, or otherwise the pathogen can spread by wind, rain, water and water splash and again infect the crops. Then the bacteria will enter into the plants through the hydatodes, and again we can see the same kind of symptoms here. Then this bacteria can enter through the roots also. Then again infect the through the vascular tissues. It will go to the leaves also. We will go to the management practices of black rot disease of crucifers. We need to purchase only certified pathogen-free seeds and also destroy infected crop residues, as the pathogen can be there in the crop residues. Then use of the crop rotation with non-susceptible crops. Then grow them in field for at least 3 consecutive years. Some cabbage, turnip, black mustard, etc. varieties have varying degrees of resistance to black rot, but it is better to grow the crops with the non-susceptible crops. Then seed treatment. Immerse the seeds for 30 minutes in water and heated up to 50 degrees Celsius. Then apply recommended bactericides. Then we will go to the last disease of Crucifers, that is cabbage yellows. The causal organism is Fusarium oxysporum. Next we will go to the symptoms of cabbage yellows. Then actually here foliage become dull green to yellowish green shortly after transplant. Then yellowish of leaves. Then all the leaves curl downward, turn partially down and shed. Then main stem become barren of foliage and often curved to one side. Then the leaves curved into one side. Young plants may be stunted and turn yellow. Then plants die before maturity. Yellow to dark brown discoloration can be seen in the water conducting tissues in the stem, petioles and veins of the leaves when they are cut through. Then sometimes Fusarium yellows often confused with the black rot. Then actually the Fusarium yellows are yellow to brown discoloration can be seen in the leaves. Here we can see difference between cabbage yellows and black rot. Then black rot you can see nicely V-shaped lesions at the leaf margins, but in the Fusarium wilt we can see yellowing of the total leaf and some particular wilting characteristics. Here also you can see cabbage yellow symptoms, then yellowing of the leaves and curling of the leaf in one side only. You can see curling of the leaves in one side only. So nicely you can see here. You can see the symptoms in these two diagrams also. Yellowing of the leaves and yellowing and drying of the leaves you can see here. Next we will go to disease cycle characteristic of cabbage yellows. Actually Fusarium fungus survive in the soil for many years as in other Plasmodivora like pathogens. This fungus can be controlled when the soil temperature is below 16 degrees Celsius. This disease is more severe at the temperature of 26 to 29 degrees Celsius. But the growth is inhibited at 19 degrees Fahrenheit. Then soil moisture and soil reaction also have effect on Fusarium yellows, and potassium deficiency leads to this infestation. The fungus invades the plant through young rootlets. The fungus directly moves towards the water conducting tissues and then progresses up the stem into the leaves. The fungus colonizes xylem tissues and does not invade the other tissues until part or all of the plant dies. The fungus produces its spores both inside and outside of affected stems. The dissemination takes place by infected beds, infected soil, farm equipments, plant refuse, water and wind. Occasionally, this fungus is carried long distances by seeds. Then actually this disease is somewhat different than the other diseases because this disease is severe at some particular higher temperature, the dry conditions, 26 to 29 degrees Celsius. And also soil moisture and soil reaction also have effect on the Fusarium yellows. If we apply the potassium with the imbalance rate, the potassium deficiency may happen. With the potassium deficiency also cabbage yellows fungus can infect. Then actually this disease is not coming from the hydrothodes or other places. This fungus directly comes from the water conducting tissues. and it will go to the stem, water conducting tissues of the stem, and ultimately to the leaves. Then actually then yellowing of the leaves can be seen, yellowing and wilting of the leaves can be seen due to colonizing the fungus in the water conducting tissues. Then water may not going into the leaves and other parts of the plant. That's why we can see the yellowing and wilting symptoms. Then we will go to the management practices of cabbage yellows. The best control method is cultivation of resistant varieties. Therefore, no problem with the management practices. Then fumigation of soil before cultivating crops is very important because the pathogen can live in the soil for many periods. Then crop rotation with non-host plants is very important, and also we need to keep the good sanitary conditions in the field, and we need to keep the proper drainage conditions in the soil, and we can use the disease-free planting materials. Up to now, we discussed the infectious diseases. Infectious diseases mean the causal organism is the pathogen, but here non-infectious disease, actually here there is no causal organism, but there may have other factors to happen the disease. The first one is cracking of cabbage. The symptom is the head burst. Then this can happen if we apply excess amount of water there. Then the control method is the harvest heads as soon as mature without keeping more time period in the field. Then the second one is poor heading. Head is not formed and or otherwise head is very small. Then this may be happen due to overcrowding of the plants, and with the dry soil, or with the high temperature, or due to poor fertility. Then the thing is, the management practices, we can use the recommended spacing for the planting and supply water, and also you all know poor fertility, then we need to apply the balanced fertilizer here. Here you can see cracking of head. Cracking of head, actually this is non-infectious disease due to excess application of water. This is actually poor heading, then head is not formed here, then here very small head formed, and here not formed the head. This is called poor heading. Now we finished diseases of crucifers. Under that we have learned infectious diseases and non-infectious diseases. Then thank you.